Welcome to this lecture on the principles of immunology. Today, we are going to explore one of the most fundamental concepts in human biology, active and passive immunity. Understanding how our bodies defend against microscopic invaders is not just academically fascinating, it is the very foundation of modern medicine, vaccines, and life-saving treatments. Let us dive in and discover how your immune system keeps you alive. To begin, imagine you are trying to protect a medieval castle from a sudden invasion. You have two distinct choices. You can either train your own villagers to fight, ensuring they remember the enemy's tactics for any future battles. Or you can hire temporary mercenaries who will defend the castle today but leave tomorrow. In the fascinating world of immunology, training your own villagers is exactly like active immunity, where your body learns to fight and remember the threat. Hiring temporary mercenaries is like passive immunity, where you borrow defenses from someone else to survive an immediate crisis. Both are absolutely critical for human survival. Now that we have our castle analogy, let us formally define these two incredible biological mechanisms. Active immunity is the state of resistance built up in an individual following effective contact with a foreign antigen, such as a virus, bacteria, or a vaccine. During this process, your own immune system actively produces specialized proteins called antibodies and activates specific cells like T lymphocytes to destroy the threat. Most importantly, active immunity generates memory cells. These memory cells provide long-lasting and sometimes lifelong protection against future infections by the exact same pathogen. Your body has done the hard work and learned how to defend itself. In contrast to training your own defenses, let us look at the mercenary approach. Passive immunity is the temporary protection acquired by receiving pre-made antibodies from another individual or animal, rather than producing them yourself. Because your own immune system is not actively engaged in recognizing the antigen, no memory cells are created. This means the protection is strictly temporary. It fades away as soon as the borrowed antibodies naturally degrade in your bloodstream. While it does not offer long-term security, it provides immediate, ready-to-use defense exactly when your body does not have the time to mount its own response. Understanding this distinction is the foundation of modern medicine and public health. Active immunity is the entire basis for how vaccines work. By teaching the population's immune systems in advance, we have been able to eradicate deadly diseases like smallpox and control outbreaks of measles and polio. Passive immunity, however, is a life-saving emergency intervention. When a person is bitten by a venomous snake or exposed to the rabies virus, there is simply no time to wait for the body to learn. Furthermore, passive immunity is the beautiful mechanism by which a mother protects her vulnerable newborn baby, transferring her own antibodies across the placenta and through breast milk. While immunology is heavily biological, we can use mathematical models to understand exactly how long passive immunity lasts. Because passively transferred antibodies are just proteins, they undergo natural biological degradation over time, following a first-order exponential decay model. This brings us to our first formula. C of T equals C0 times e to the power of negative KT. Here, C of T represents the concentration of antibodies remaining in the blood at a specific time t. C0 stands for the initial concentration immediately after they are administered. The variable K represents the elimination rate constant, telling us how quickly the body clears the antibodies. If the initial dose increases, protection lasts longer. If the elimination rate increases, protection disappears much more quickly. To find that elimination rate constant, we often rely on a more intuitive clinical measurement, known as the biological half-life. The half-life is simply the amount of time it takes for exactly half of the circulating antibodies to be degraded and removed from the body. The formula is T1/2 equals the natural logarithm of 2 divided by K. The symbol T1/2 represents the biological half-life, usually measured in days. The natural logarithm of 2 is approximately 0.693, and K remains the elimination rate constant. This formula tells us that half-life is inversely proportional to the elimination rate. If the body clears the protein very rapidly, the half-life will be very short. For example, IgG antibodies typically have a half-life of about 21 days. Let us see this in action with a concrete example. A patient is bitten by a venomous snake and receives an injection of antivenom. The initial concentration is 120 milligrams per liter, and these antibodies have a half-life of exactly 15 days. We need to calculate the concentration after 45 days. First, we determine how many half-lives have passed by dividing the total time, 45 days, by the half-life of 15 days. This gives us exactly 3 half-lives. Next, we use the discrete decay formula, multiplying the initial 120 by 1/2 raised to the power of 3. This perfectly represents cutting the concentration in half 3 separate times. After performing the arithmetic, we find the final concentration is 15 milligrams per liter. The patient only has 1/8 of the original protection left, proving passive immunity is strictly temporary. Now that we have calculated the decay of passive immunity, let us shift our focus to active immunity. While passive immunity inevitably decays, active immunity is measured by how well it prevents disease in a population over time. In clinical trials for new vaccines, researchers observe the incidence rate of a disease in an unvaccinated control group and compare it to the vaccinated group, whose immune systems were actively trained. To determine exactly how well this active immunity protects the population from the virus, we use a standard epidemiological metric known as vaccine efficacy. This metric quantifies the protective effect induced by vaccination by finding the proportional reduction in disease incidence. This brings us to the mathematical representation of active immunity. The vaccine efficacy formula is VE equals 1 minus the ratio of IRV over IRU, all multiplied by 100. In this formula, VE stands for vaccine efficacy, which is ultimately expressed as a percentage. The symbol IRV represents the incidence rate in the vaccinated group, while IRU represents the incidence rate in the unvaccinated group. By dividing the vaccinated rate by the unvaccinated rate, we find the relative risk of getting sick if a person is vaccinated, compared to if they are not. Subtracting this relative risk from 1 gives us the exact fraction of risk that was completely eliminated by the active immunity. Let us apply this formula to a real clinical trial scenario. Researchers observe an incidence rate of 0.050 in the unvaccinated control group and an incidence rate of only 0.005 in the vaccinated group. We substitute these given data points directly into our efficacy formula. The vaccinated rate goes in the numerator, and the unvaccinated rate goes in the denominator. The ratio of these incidence rates is 0.10, meaning vaccinated individuals have only 10% of the risk compared to unvaccinated individuals. Subtracting this value from 1 leaves 0.90. Multiplying by 100 gives a final vaccine efficacy of 90%. This physically means the active immunity successfully prevented 90% of the infections that would have otherwise occurred. Let us visualize everything we have learned by looking at this graph comparing antibody concentrations over time. On the horizontal axis, we have time in days, and on the vertical axis, the antibody concentration in the blood. Notice the red curve representing passive immunity. It starts at its absolute maximum the moment the antibodies are injected, but immediately follows a steep exponential decay, eventually fading to zero. Now, look at the blue curve for active immunity. It starts at zero and takes time to build during the primary response. However, upon a second exposure, the memory cells trigger a massive secondary response. The antibody levels skyrocket and remain high for a very long time. This diagram perfectly illustrates the trade-off between instant temporary defense and delayed long-lasting protection. We have covered a lot of ground today, so let us summarize the most important ideas. First, active immunity requires your body to do the hard work of recognizing pathogens and building memory cells, which results in delayed but incredibly long-lasting protection. Second, passive immunity provides instant, life-saving defense by borrowing antibodies from an outside source, but it leaves no memory and fades away quickly following exponential decay. Finally, we saw how mathematical models allow us to calculate the half-life of borrowed antibodies and the efficacy of vaccines. By mastering both of these concepts, medical professionals can choose the perfect tool, whether it is a vaccine for the future or an antivenom for today. You now have a solid understanding of how immunity works.